I'd like to introduce my friend and colleague, Elizabeth Blackburn.
Hello. In this session, we're going to celebrate two remarkable scientists who've played pioneering roles in the fields of genetics and genomics, which, because of their visionary work, really have become almost indistinguishable fields. And I was thinking about this historic sweep of what their work has represented. And I was thinking about Fred Sanger, who at the age of 95 just died just a few weeks ago. And he, of course, pioneered the DNA sequencing methods that made the human genome possible. And he was just so amazed and delighted at how things developed so quickly. And in 2001, when the human genome came out, he said, "When we started working on DNA," this is Fred Sanger speaking, "I don't believe we were thinking about sequencing the entire human genome, perhaps in our wildest dreams, but certainly not within the next 30 years. Well, it's our speakers today whose vision and real extraordinary accomplishments made this not just a wild dream, but into a reality. And so, looking at the sweep of this, we've gone from, the DNA sequencing to the huge amount of extensive and, deep knowledge that you get from genomic sequencing. And you look forward now into, I think, what that in turn is going to be bringing, which is going to be, incorporating that and greatly enriching much greater, wider even regions of human health and disease and in fact, profound insights, I think, into biology in general. So David Botstein and Eric Lander are the two fantastic scientists who are being celebrated today and in this session. And first of all, David's going to talk on yeast, evolution, and cancer. David.
I think I'm all set. Thank you, Liz. It's, of course, a great pleasure to be here. It's a great occasion. And it's nice to be back at UCSF. I was on sabbatical here last spring, and so I got to know the place and this room pretty well. And anyway, thanks for everything. My talk is going to be really a change of pace, because I'm a basic scientist. I'm a geneticist. I'm not a cancer biologist. I'm not a physician. I don't do any of those things. But what I do is think about genetics. And what I'm going to tell you is a story about evolution. And I don't work mainly with animals. I've never killed a mouse. Okay? Never made it sick or anything like that, okay? That's not what I do. I work with yeast. Now, yeast are the premier model system for eukaryotic biology. The great take-home lesson of the genome sequencing was that the basic plan of everything from metabolism to everything except the cell wall is basically shared by animal cells and yeast, except yeast are much more efficient and have a lot less overlapping complications like Lou Cantley told you about. But the central part of Lou Cantley's picture is there in yeast. Tor, as he said, Mike Hall, found Tor in yeast, and he took advantage of that relationship. And in fact, all of those, I know all of those genes by different names. In fact, because the animal guys always change the names, okay? And Tor they didn't change, everything else they changed. But in fact, some of them actually were discovered in my lab as mutants, okay? We didn't have no idea that I'd do anything with cancer, but of course, the general belief in the medical community is that scientists like me, who are just trying to figure out which end is up, won't notice when something comes up that's useful for cancer or something like that, that will suppress it somehow, and that we need to be goaded into translation. Okay? And I'm here to tell you a story which says that maybe this enthusiasm, this stampede to translation, is maybe just a little bit misguided. That we still have a great deal to learn about the basic biology that will inform the kinds of studies that we really need in order to understand what's going on in things like cancer, and in fact, especially cancer. So, fundamental result of genomics, as I just said, is that everything seems to be extremely conveniently conserved. Nature did us a huge favor by having the rate of evolution be so slow that we can follow a protein sequence back to the common ancestor of all eukaryotes if that protein sequence is important enough to cell biology. So we've learned a great deal from yeast, and in fact, as I said also before, if you look in the databases and you look carefully, you'll see that about half of all the annotations of human genes lead back to yeast, okay, or Drosophila. That's the one thing that happened in the last decades. The other thing is that there emerged a consensus, which you heard brilliantly explicated here this morning, that cancer is the result of serial mutations, somatic mutations, which eventually end up with something that isn't doing what it was evolved to do for the organism, but is doing for itself, and it's going to kill you. Okay? The question is, if the parts are conserved and the interactions are conserved, maybe the mechanisms of evolution are conserved also. So we're going to think about evolution, first in yeast, and then occasionally maybe not in yeast. Okay, the story for me starts with these guys, Jacques Monod and Leo Szilard. Most of you know who it was, Monod. Leo Szilard, maybe not so much. He was a minor 20th century physicist whose main claims to fame were he invented the chain reaction, sensu reactor, neutrons, stuff like that, and the atomic bomb, okay? And he got patents on both, okay? However, at the end of the war, he turned to biology, and one of the things that he and Monod, in those days people did much more collaboration on basic ideas in public than you see now. They invented this gadget, which Szilard called the chemostat. And what the chemostat is a way of growing free-living bacteria or yeast in a vessel which you're constantly dripping in fresh medium, you're dripping out finished culture, and what this means is that every time a drop comes in here, drop of finished culture comes out, and you can easily see that under some conditions it must be that you get a steady state, that the dilution here is exactly matched by the growth. Now, it turns out that that's really true, and all organisms that have been tested in this system have the property that they adjust to their nutritional environment, which is, the unspoken assumption of Cantley's talk, that they adjust to their nutritional environment if they're not sick, and in a way which is Tor and all those things have a lot to do with. Okay, so in this thing, this is great for systems biology and stuff like that, and for the mathematicians, because steady state means that all the derivatives of all the equations that run anything are zero. Okay? And so this is the situation you want to be in when you want to calculate something. But also, it turns out that under these circumstances, I can tell you why the culture is growing at the rate that it's growing, what is the physiological reason, as well as the reason that I changed the flow rate. Because I can arrange it to know what the limiting nutrient is. I can limit them on glucose, I can limit them on phosphate, I can limit them on anything I want. And the second thing is that under those circumstances, there's a primary selection. There's a selection for guys who can better use glucose, or better use phosphate, or better use sulfur, whatever is limiting. And so this is a machine which you can run for months, and you can look at thousands of generations and ask, do things arise which grow better? And, does the bear do it in the woods? Of course things arise which grow better. And this is what we are pleased to call experimental evolution. Okay? If you do this with glucose limiting, you get a very pleasing result, which is that it turns out that the evolved yeast have read Streyer's biochemistry book. In Streyer's biochemistry book, it says that if you take glucose to alcohol, which is yeast's preference, they get 2 ATP per molecule of glucose. And if on the other hand you go through all this nasty oxidative metabolism, then you get, well, it depends on which edition of Streyer's book. It's somewhere north of 30, okay? But every edition gives a different number, okay? So this hasn't been worked out yet exactly, okay? And what you see is that the evolved strains have turned down these genes, green means down, and have turned up these genes. In other words, they have shifted metabolism from fermentation to oxidation. We did this 3 independent cultures, got the same result. Here's the data. I'm going to remember CYT1. And then here's the data. You see that the respiration genes are up, the fermentation genes are down, and the transporters, like politicians, are divided. Okay, if you do this in diploids, you get the same kinds of results, except what you really see in diploids is if you measure copy number of genes, you see that you get changes in copy number. Over here, you see the hexose transporter has a copy number of north of 8. Okay? So somehow the yeast have adapted by increasing the copy number of the hexose transporter. Makes perfect sense. So far, we're not in the dark. We're still doing good. And then we have reproducible translocations. The translocation here, translocation here. There's a not translocation, but same breakpoint there. And those of you who have been listening this morning will suddenly realize, wait a second, this is what happens. I'm going to skip a little bit. This is what happens. I'll come back, don't worry. This is what happens in the Philadelphia chromosome. Exactly that. You reproducibly get, for the CML, you get this translocation between BCR and ABL. Okay? In that case, it's a hybrid protein, but it's not entirely clear whether it's the protein or its expression that actually matters. So if I go back, you can see that in my case, what we got is a translocation right here at this point. Data are really good. This is the karyotype of yeast. I won't bother you with that. I'm going the wrong way. Okay. And what you see is that break is right here at this transposon remnant, and there's the data again. And then if you look at where is that, it's CYT1, and you will recall that CYT1, okay, right there, it's right at the promoter, is citrate synthase. And what is that? That's the first step of the oxidative pathway. We can understand that one too. Now, if you do this now on a larger scale, you buy more chemostats, you have more postdocs, you do more experiments, you get, and now you use different limiting nutrients. What you get is this kind of thing. So what we're doing here is we're looking at the total patterns of expression. These are the genes. These are the various evolved strains. We just fish one out and measure it. And what you see is that there are several ways to adapt to glucose, phenotypically. This is a phenotype. There are several ways to do a phosphate, and there's maybe 1.5 ways to deal with limiting sulfur. And in fact, this is a fancier way of looking at the same thing. There's one peak for sulfur. It's more complicated for these other nutrients. This is a very important point. There's only a limited number of ways that the yeast can actually improve its condition in the chemostat. It has to do something about its limiting nutrient, and the way it's wired and the way its system works, there's only a few things it can do. And what we're doing is we're sweeping these out. But chemostat by chemostat, it's going to take us forever to figure this out. So I'm going to switch to something else. But before I do that, I want you to consider tumors, because Pat Brown and I spent a decade looking at tumors by the same technology of gene expression. And what you see here is all different kinds of tumors, and they're color-coded by the type of tumor. And what you see is that the microarray platform is never confused about whether a breast tumor is a breast tumor. Another way of looking at that is to say the breast tumors are never confused about where they came from. They're still breast epithelial cells. They may be de-differentiated in some trivial sense, but they're not going to get confused with ovarian cells. However, and each different cell type has its own issues, just like each limiting nutrient is different, because each of cell type is in a different environment. Okay? So, however, there are subtypes. And these subtypes, okay, turn out to be significant. This is one experiment, and this is the same thing done on somebody else's data. You see all the red guys are together, all the dark blue guys are together, all the light blue guys are together, and we didn't have enough magenta guys to be significant here. And in fact, their clinical outcomes are also different, and this is very robust to all the technical and biological changes between, let's say, Holland and Norway. Okay. Now, going back to yeast, sulfur, you remember, was unique. All the sulfur ones were similar. And if we look at gene copy number changes, they all start in the same way. They all start down the same path. And that path is amplification of the sulfur transporter. And here you can see data. And now I go back to the breast cancers and I ask the following question, which is really driven by the same idea behind the sulfur thing. If there are 4 different kinds of breast cancer, then women who inherit a head start, they should be going down one of those paths and not all 4. Okay? That's not a logical requirement, but it is of high likelihood that that's the case, as it was with the sulfur transporter. And so we can test that. This is a genomic hypothesis test. We can ask the question, which type are the people who had BRCA1, because BRCA1 is almost 100% by the end of normal life that you'll get a breast cancer. Okay? And what you see is BRCA1 carrier, all one subtype. They're the what is now called triple negative, but which we call the basal-like subtype. We didn't have enough data to deal with the BRCA2 carrier, so ignore that. Here's another thing. Now I'm going to go away from what we already know to maybe things that you don't know that we learned from yeast. So if you limit on nitrogen, yeast care a lot about nitrogen. Nitrogen is a really big problem, has been a big problem for them evolutionarily. And so they can use many different nitrogen sources. Among them glutamine, glutamate, allantoin, which is a purine degradation product. And the remarkable thing is that if you look at copy numbers, well, glutamine, you get amplification. Glutamate, you also get amplification. Allantoin, you get deletion of this particular same gene. What is that gene? That gene is called the general amino acid permease. And if you're doing allantoin, having the general amino acid permease seems to be a problem. But if you're doing glutamine, it's an advantage to have lots of it. Okay? There's also a mechanistic clue here, because there are 2 transposon remnants that are facing the same direction, and the sequence, and there's a autonomous replicating site right there. And so the possibility exists that you can get a circle. And in fact, it turns out that both these deletions and those amplifications are start with this internal recombination that gives you the circle. There are 231 transposon remnants in the yeast genome. In human genome, there are, 100 million or something? Very large number. 2 million, okay. Anyway, way more than 231, right? Okay. Way more than 231. But the ones in yeast, if you go and look through the yeast evolution, they are all still there. They have not moved around. Okay. Of course, this kind of amplification, both local and disperse, is characteristic of certain kinds of tumors, and it's particular kinds of tumors that have particular things, suggesting very strongly that the local genomic situation, the propensity to recombination, transposons, whatever, is relevant to which kind of amplification mechanism is most commonly recovered in the tumor cells. Now a little theory, okay? And this is totally relevant to this business of how many mutations and so on. We're now going to talk about how many different point mutations. Okay, so Michael Desai was a fellow at Princeton, and his thesis advisor, the famous Daniel Fisher, they wrote a paper. And in this paper, they made what seems in retrospect like the obvious point, that if you have a fitness along this axis, and you imagine a small population, and you have a large fitness mutation, that what will happen is that this will take whatever lucky cell got that mutation and give it a head start, which it will outrun the population with high probability. If, on the other hand, there's a lot of standing variation already, then it matters who is the lucky cell. If the lucky cell is one of these living dead, the best it can do is get to the nose of the distribution. And it may or may not prevail. But if it was one of the ones that already is at the nose of the distribution, then it will prevail with high probability, just as this one. Now, what would bring it to the nose of the distribution? Well, you could say it was epigenetics, you could say it was luck, you could say, or it could be another mutation that it already had a fitness mutation. So this idea can be tested. So what we did was, this is all for the yeast people. There are many, many genes which if you knock them out, you grow faster if you're a haploid. And the reason these things are all involved in sex, and if you're a haploid yeast and you're not looking for sex, then you save energy. Okay? That's really what it comes down to. All right? And we can hook up the sex to a GFP, and we can use the fluorescence activated cell sorter, and we can detect 5% of one type in the presence of the other. And now we can ask the question. We have an expectation. We have a large fitness mutation that we know can happen, which is inactivation of any of these 5 genes. What happens? Well, we look at over 1,000 generations, now we're no more 24 chemostats, in 600 independent cultures. And what you see is that, sometimes a sterile mutation comes up and sweeps the culture. Sometimes it's more complicated. It looks like it's going to win, and then it got overrun by a competing mutation. The theorists had expected all of this stuff. Sometimes it's more complicated than that, and sometimes you get new kinds of things, which I'm not going to pursue further here. The important point is that Desai and Fisher said, well, you had to have a boost before you could actually take over. And so we measured the slope, the rate at which these guys are outgrowing the population, versus the fitness advantage of the mutation. And what you see in every case is that the reality is that it's not just the mutation. There's something else. Okay? They actually are growing faster than they should, all of them. Even this one, who for the population geneticist has just undergone purifying selection. Right? Just to remind you of the theory. So we said, what happens if we sequence the whole population to very great depth? Are we going to find in here a mutation, or is it some epigenetic, who knows, mumbo jumbo? Okay? All right? And so it could easily be, I mean, this is a way of detecting epigenetic something or other, okay? But in our case, what happened was that there were two mutations, ELO1 and GAS1. And when we read the annotation, yeast is very well annotated. Fatty acid elongation, that sounds good. Chromatin silencing, that sounds good too. Could be either one, could be both, could be neither. Okay? The sterol, which is a nice frame shift, okay, was what we expected, right? So, this is not tumor biology. This is yeast. We can figure this out. We can do what the ancients called a backcross. And when we do the backcross, okay, what we see is that here's the ancestor, here's the evolved, here is the backcross. What we see is 1 to 2 to 1. And all we have to ask the question is, are there GAS1 mutations in here and in here and in there? Are there sterol 12 mutations in here and here? And the answer is that GAS1 is the driver, and this other mutation, ELO1, with the fatty acids, is neutral. Okay? And together it all adds up. Okay? So we can do this enormously deeply. We have 600 of these. We've fully sequenced 60 of them every 80 generations, and we get all kinds of dynamics of evolution, and most of these dynamics I can point to a cancer case where the same thing must have happened. Okay? All kinds of things. One of the very interesting things, we get a lot of passenger clusters. And maybe they're not just passenger clusters, we haven't worked them all out. Maybe some of them are like the PI3 kinase, that you can't actually get there with one to the full extent, and you need more. And here is the part that I like the best. This is why we did this experiment as far as I'm concerned. Ask the question, well, is it always the same genes that allow you to grow better in this microtiter well growth system? Well, we can ask, how many times did we get the same gene? Well, it turns out that IRA1 and IRA2, we got a lot of times. And IRA1 and IRA2 are regulators of RAS. Does that sound familiar? That was part of Cantley's diagram right there. They're called something else in animal cells, but it's the same RAS. Okay, they didn't change the name of RAS, thank goodness. Actually, that's because we found RAS and we didn't change their name. Okay, because we found RAS after they had found RAS. Okay. Yes, non-reciprocity. Okay. We also find other things. Mating, of course, we expected. We also find all these cell wall assembly. And so it turns out, with evolution, you can't fool Mother Nature. So it turns out that in these, pedal to the metal growth circumstances, what seems to be limiting is making a proper cell wall. Think 3 little pigs. Okay? You want a brick house, not a sticks house or a straw house. But if you're in a big hurry and it doesn't matter, you'll make do with the straw house. So basically, all of this complicated cell wall stuff gets short-circuited and they grow faster. And how do I know this is true? Is because if I pick one of these things out and I try to grow it under other conditions, It hates those other conditions. They lyse. They're hypersensitive to God knows what kind of, minor drugs, pH, and stuff like that. They're not fit, except in the circumstance which we actually selected them for. These are all negative regulators, and you can see these cell wall guys, however, turns out that the chromatin is a minor point. It's a cell wall gene. It's actually an enzyme. And it turns out that here we get point mutations, and here we get loss of function. So molecular biology works. All right, so the conclusions are there are striking similarities between the genetic events. You have to think of these as continually evolving populations. Frequency of occurrence, I didn't talk about this, skip that. Number of paths to fitness is limited. There's not 100 ways to get better. There's a limited number of ways, which we've certainly seen in most cancer settings. And dysregulate, I didn't talk about this, but this kind of dysregulation that Lou talked about also happens in yeast, but under different conditions. And the Warburg effect is alive and well in yeast, and so on. And with that, I will stop and thank everyone again, and show some of the people who did the work. Most notably Maitreya Dunham and David Gresham, who did the hemostat experiments, Greg Lang and Desai, who did the point mutations. And thank you.
Wonderful. Questions for David? Lou from the front row.
Just a comment. Warburg called it fermentation.
Yes, he did.
He didn't call it the Warburg effect. He called it fermentation.
Right. Yeah. Yes.
You didn't see RAS mutations. Does that, do you think, because it's easier to inactivate R1 than it is to activate RAS, or is there some biological difference?
I actually think that the actual RAS mutations are so poorly fit, they're so intolerant to going into stationary phase that I think that in the way it wasn't continuous culture, and so I think that they were selected against every generation because they don't like they have additional phenotypes.
But
if it was your implication that different subtypes of breast cancer derive from different evolutionary solutions, which I sense was your implication, it may not be correct because it's almost certain that the different subtypes of breast cancer derive from different normal cell types, where the normal cell of origin's differentiation program continues to impose itself on the cancer cells.
It was your first implication. I did not mean that. What I meant was that the genetic pathway by which they become independent of whatever growth control, they have several options, and the one that they choose predisposes what happens later. Okay? I did not mean to say that they're in different environments or that they're different cells.
Cynthia.
If you just think kind of, I don't know, a priori, you might think if you just let yeast evolve, you could get anything. But you don't. You just get a few different... So the initial starting conditions really constrain the outcome. And the question I have is, if you start with bacteria, or something that seems really different from yeast, if there is such a thing, maybe some archaea or something, can you get new solutions?
Well, I think that my lifetime is limited. My postdoc's lifetime is also limited. Whereas the bacteria have had, a billion years to work this out among themselves. And so I think that they're limited, and the species of bacteria that you see are the different solutions. And I think that if you, that what you can do is you can get a bacteria then to get into some specialized situation and grow better, but then they will lose things that they would have. So for example, our mitochondria are derived from some bacteria. But in order to become really good mitochondria, they have removed all but about a dozen proteins from their own genome, right? So this thing is not a free-living anything, right? Okay, you take a mitochondrion out, you have to work very hard to keep it alive, and I don't know if anybody can actually grow mitochondria in vitro. I don't think so, okay? Maybe somebody knows, but I don't think so. And so I'm really thinking that in terms of the evolutionary time, we're so far down the road that I don't think experimentally what we can do is see anything except the beginning steps.
Great, thank you. And unfortunately, our time sadly is limited. So thank you very much, David, and great talk.